So, good morning. So, in the last class, we have discussed about the heredity. Okay, and there are some terminology we have discussed about what is gene, what is allele, what is dominant trait, what is recessive trait. Okay, what was the role of Gregor John Mendel in the genetics? Then what is the term genetics? Then what is chromosomes? What is genes and the difference between DNA and genes? Okay, so these were the basic things we have discussed about the heredity. Okay. Then pod color, green, yellow. Then flower position, where is the flower is located? Either it is on axillary portion or it is on the terminal portions of the stem. Then what was the stem height they have taken, like tall, dwarf. So they have he has taken the some seven contrasting characters. He have seen the seven contrasting characters of the piece, and he has selected accordingly. So after then we have seen what was the monohybrid cross. So in monohybrid cross, what we have seen. So first, what he has done. Okay. So he has taken the pea plants for his research purpose, that is Pisum sativum. So what he has done, first he have done the monohybrid cross, and after then he has done the dihybrid cross. And what he has done in monohybrid cross, first he has taken the one contrasting character, that is the stem height, that the height of the plants. So what he has done, first he has taken the one tall plant, after then he has taken one dwarf plants. Okay, these are the homozygous allele. Okay, what is homozygous, heterozygous? So if the allele is same, it is the same even in the opposite, that is called homozygous. Heterozygous, if it is different, that we can say the different allele. Okay, that is called heterozygous. So in homozygous, he has taken the one tall plant, after then he has taken one the dwarf plants. So after then he has crossed the one tall plant and one dwarf plant. After then, he has seen, observe that if it is a parent generation, if it is parent generations, so in the F1 generation, what you have seen, the 50% character of the both the parents material has been transferred to the next generations. Okay. Then we have seen also the phenotypic term and the genotypic term. Okay. So phenotypic is generally the outer appearance, the outer observation of that plants, that is phenotype, and genotype is that it is the genetic makeup of that plant, that is genotype. So after the crossing of the parent, that is tall plant, and the dwarf plant, he observed in the first generation, after the crossing he got that F1 generations, that is capital T small t, the genotype he got, that is capital T. In the tall, from the tall plant, the capital T, the dominant character, that is the capital T is transferred, and from dwarf plant the recessive, that is T was transferred. But in the F plants, after the crossing of the F1 plants, he got the ratio of 1 is to, he got two tall plants. He got two tall plants. Like if we talk about the genotypic ratio and phenotypic ratio, so in F1 generations, he got that genotypic was that is 1:1. Okay, and the phenotypic was 2, because the whole, whatever the plants he got in F1 generations, that is everyone was tall. Okay, but their genetic makeup was is capital T small t. Okay, so he found that the phenotypic what plants in F1 generation he found that was everyone is tall. So after then he has done again the crossing. After then the what he taken the F1 generation that is capital T small t. After then he has taken the F1 generations. After then he has again the self pollination, the self crossing of that F1 generation in that generation only. So what he found in that found that the capital T, he found that four types of genotypes, and the four whatever the plants he found in F1 generations, that was the in monohybrid cross, that is capital T small t. After then capital T small t, then small t. Okay, so that is the mono hybrid cross that is found between the F2 generation. So in F1 generations, so from the first parent generations, first he has taken the tall plants, then dwarf plants. After then he found F1 generation that in capital T small t. In F1 generation, all plants were tall. Okay, and then genotypic ratio is 1:1. Okay, only one genotype, but the same genotype. After that, he again does the crossing between the F1 generations. So he found four plants. Okay, in four plants, the genotypic ratio was 1:2:1, and the phenotypic ratio was and the phenotypic ratio was and the phenotypic ratio was 3:1. So after the crossing of one F1 generations, what he found that the genotypic, that is, he found the two homozygous different allele, and he found one heterozygous, that is two heterozygous he found. So he found one tall homozygous trait. After that, one homozygous recessive trait. After that, he found two heterozygous trait. Okay, so that is after the crossing of F1 generations in the monohybrids. The Mendel found the F2 generations. He observed that whatever the character that was present in the parent that was not expressed in the F1 generations, but in F2 generations that character is expressed in the recessive trait. Like that small t that was the recessive trait that was not observed in that was not expressed in the F1 generations, but after the crossing of F1 generations in the self crossing, he found that the recessive trait is also expressed in the F2 generations. Okay, so he found that two plants were tall, the three plants were tall. Okay, and one plants were recessive, and he found that the character of the parents that was already transferred in the F1 generation, but that was not expressed in F1 generations, because that dominant trait is dominated to the recessive one, and the recessive trait is something that not able to express itself, and the dominant trait is the trait that suppress the expression of the recessive one. So that is a dominant trait that was already transferred on the parents from the F1 generation to the F2 generation, but that was not expressed in the F1 generations. So that's how Mendel found the genotypic ratio and the phenotypic ratio was. In the genotypic ratio, he found that is 1:2:1 ratio, and the phenotypic ratio is found 3:1, that the three plants were tall and the one plants were dwarf. But in F1 generation, he found the whatever the plants he observed or the got, that was already everything was tall only. Okay. So Mendel selected the garden pea plant Poison Satyr for his experiment. He selected tall and dwarf plants and allowed them to grow naturally. As pea plants produce seeds only by self-pollination, he observed that tall plants produce always tall plants generation after generations under natural conditions. So what we have discussed. So similarly, dwarf plants produces always dwarf plants generation after generations. Hence the term the tall and dwarf plant as wild types or pure breeding varieties. So then he again crossed a tall plant with a dwarf plant, and which has produced progeny, and calculated the percentage of tallness and dwarfness in subsequent generations. When a pure breeding tall plant was crossed with a pure breeding dwarf plant, all plants were tall in the first filial generations, that is F1 generations, and there was not any medium height plants or dwarf plants. But in this means that only one of the parental traits were seen, and not the mixture of the two. When such a F1 tall plant was allowed to have self-pollinations, both the tall and dwarf plants appeared in second filial generation, that is F2 generation, in the ratio of 3:1. This indicates that both tallness and dwarfness were inherited in the F1 plants, but only tallness trait was expressed. The first experiment of Mendel, considering the inheritance of a single trait, that is the height of the plant, that is tall or dwarf, is called monohybrid cross. And the expression of morphological characters as tall or dwarf plants, violet or white flower, that is called phenotypic expressions. The expression of gene of an individual for a particular is called genotype. So this is how that the experiment setup was confirmed, and that was predicted by the Gregor John Mendel. So first he have taken the parental, that is tall plant, and the dwarf plant. After that he crossed between the tall and dwarf. He got the one F1 generation. Whatever the progeny he got in the F1 generations, that again that F1 generation is went for the selfing. In the selfing, in the F1 generation, see the both plants were tall. But after selfing of that both plants, he found that in F2 generation, the filial two generations, the tall, two tall, three tall, and the one dwarf characters. So these were the diagrammatic representations of monohybrid cross that was done by the Gregor John Mendel. Then what are the types? The heredity we have discussed, that is the passing of traits from the parents to offspring, is called heredity. Then what is genotype? The complete set of genes in an organism, that is genome, is called genotype. Then what is the phenotypic? The observable character in organisms make the phenotypic, and the phenotype is a result of genotypes interaction with the environment. Due to this reason, many phenotypes are not inheritable. Then acquired traits we have also seen, like what I talked about the last class in the South Korea example. Then interaction with the environment, that is acquired traits. Then inheritable traits, that is transferred from generation to generation, subsequent generation, that is inheritable traits. Then Mendel's first law. So we are discussing about the some there are some laws were proposed by the Gregor of Mendel. The first law was law of segregation. So what is the law of segregation? It means whatever every individual that will possess a pair of alleles for a particular trait, during gamete formation, a gamete receives only one trait from the alleles. A particular trait can be dominant or recessive in a particular instance. For example, segregation is something like if the capital T, for example, we are talking of the F1, F2, F2 generations, in we have found there's one heterozygous trait was there, capital T, small t. Okay. So what will happen? So if we are crossing between these two heterozygous, what we are getting? We are found the four alleles. We found four alleles: the capital T, the small t, the capital T, a small t. Separate. We got four. Alleles. So what happened when after the crossing of this, the ratio we have found, the progeny we have found after the segregation. See what happened? The allele was present instead of the allele was present in the pair, but they when they were when after the crossing, what happened? The 50% of this material and the 50% of this material were get transferred, and that was selfing. That was producing one another individuals. For example, if the capital T. So this is how every individual poses a pair of alleles. Every individual has a pair of alleles. Like they have pair of alleles, like they have homogamy, they have they have pair. Like this one is also one pair. That is also one pair. This one is also one pair. Okay, so we have heterozygous and homozygous. Okay, so every individual possesses a pair of alleles for a particular trait during gamete formation. So after the selfing, after the crossing, so during gamete formation, a gamete receives only one trait from the alleles. So after the crossing, they are not going to receive the both alleles. They are that is going to receive only one allele. That is either they will receive capital T or either they will receive small t. So from after the split, they are going to attain only one allele. So one trait from the alleles, a particular trait can be dominant or recessive in a particular, or that allele can be dominant or that trait can be recessive. For example, in the case of F2 generations, we have found the four individuals, that capital T, capital T, capital T, small t, the capital T, small t, and the small t, small t, in which we have found there was four alleles. From the four alleles, the separate gamete formations. In that formations, they have taken one gamete from the uh, that in F1 generations we have taken the capital T, small t, and we have crossed. So from the two parent, that F1 generations, he has taken the dominant one, that capital T, capital C. So because of that, these plants were tall. Again, he has taken the capital T and the small t, and because of that, the capital T was dominant to the recessive one, and he expressed the and it expressed the character that was the tall. And again, in the second also, they have taken the dominant and the recessive one trait. So we have seen there is one segregation is there. It's not like two alleles is coming together and forming the gamete. The individuals of the gamete, they are separating. They have pair of alleles, but only one gamete is going for the formation of the character, and only in the gamete only one trait is going to form an organism. So a particular trait can be dominant or recessive in particular generations. So these all are about the monohybrid cross, whatever given by the Mendel's. Then let's talk about the dihybrid cross. Okay. Let's talk about the dihybrid cross. So the cross in which two pairs of characters are studied is called dihybrid cross. In his second experiment, Mendel used dihybrid cross. See, in monohybrid cross, what we have seen in monohybrid cross, we have just taken only one trait. We are Mendel was studying only one trait of the character, that is tallness or dwarfness of the plants. He has taken only one contrasting character, that is the height of the plants. Okay, in height of the plants, he has taken the tall is the dominant one and the recessive one is dwarf. Okay, but in the dihybrid cross, we are going to take two contrasting character of that plant. Okay, so that is dihybrid cross. So the cross in which two pairs of characters are studied is called dihybrid cross. In his second experiment, Mendel used dihybrid cross. So in the first experiment, he has taken monohybrid cross, F1, F2 generations, and he has seen that he has observed the 1:2:1 for the genotypic ratio of in the monohybrid cross, and the in phenotypic he had found the 3:1, that is the in monohybrid cross. But in dihybrid cross, we are going to cross two contrasting character of that plants. Okay, so that is dihybrid cross. Okay, so let us take an example between plants with round and green shapes. Okay, so we are going to take two characters here. That is between plants with round and green seeds. Okay, we'll take the two contrasting character, that is the shape of the seed and the shape we are going to take two characteristic character, that is the seed color and the seed shape. So seed color, what was that? That was yellow and green, in which we have found the yellow one dominant one and the green was recessive one. Again, in the seed shape, we are going to see the round and wrinkled one, in which we have seen that dominant one was the round one and the recessive one was wrinkled. So we are going to discuss these things in this dihybrid cross. So let us take an example. Here he has, you can see here. So he has taken the Alice. So he has taken two characters. So how? Okay. Okay. See, he has taken. He was doing the dihybrid cross. In he see the genotype of round and green seeds is shown by RRY, and that of wrinkled and yellow seed is shown. Okay. I'll take example here. See, two traits he has taken, in which he is going to find a lot of knowledge. So we'll just form the arrays like two characters he has taken. Like for the capital, okay. So here we are going to take two characters of that allele. Okay. So we are going to take about the round Okay, four alleles, there will be four alleles will be formed. Like we have uh, uh, two characters of we have two traits of each character. So if there is two character, we have to two contrasting character, then we have to take the four traits. Okay, so round yellow, then round green, then we have wrinkled yellow, then we have wrinkled green. And we already know that the dominant in if you talk about the seed shape, in which the dominant was dominant was round, and the recessive was wrinkled. Okay, again in the seed color we know that the dominant was yellow, and the recessive was green. Actually, Mendel was working on this study. So after the dihybrid cross, he got this one is dominant and this one is SS. But we already know. So how Mendel worked on that? So he has taken the he has taken two characters, that is seed shape and the seed color, in which the he after the experimental setup there his observations, and he found that dominant was round one and the recessive was wrinkled one after the observation, not before that. Before Mendes, nobody knows that which one is dominant, what is the recessive trait. Even this terminology was not developed on that time. Okay, so what he has taken, so in for dominant, he has used the term for the dominant. He has used the term that is uh, capital R, and the for recessive he has a small r. Like that in the seed color, he has taken that y for the yellow one and the small y for the green one. Okay, after then he formed the after then he has done the diagnosis. So now we can form here the round yellow. So if there will be round, so r, and for the yellow y, r, y, then small r, y, then small r, y. So there is four. So that was the dihybrid clause. Now what happened? We know that law of segregation will happen. Law of segregation will happen or not? Definitely it will happen. So what will happen here? Capital capital R, one y will be segregated. Capital capital R, one small y will be segregated. Small r, then small r, capital Y will be segregated. Then a small r, uh, small y will be segregated. Okay, it will be segregated. Now we will do the crossing between these things. Okay, so what will happen now? Okay, so what will happen now? We will take one box. Okay, now we'll form some lines. One, two, three, four. Okay, okay, okay. So what will happen now? We will form the values. Capital R, R, small r, small r. After then, what will happen? Capital Y, small y, capital Y, small y. Okay, so this one was capital R, capital R. These are capital R. Then again, uh, small r, capital R, small r, small r, small r. Again, this one is capital Y, capital Y, capital Y, small y, then capital Y, small y, capital small y, small y. Okay. So after monohybrid if then what will happen? Capital R, capital R, capital Y, capital Y, capital R, capital R, capital Y, small y. Let's see if it is given in there. Okay. Okay, we'll just continue in that way. So then capital R, capital Y, small y, then capital R, small y, small y. Then again capital R, capital Y, capital Y, capital R, small r, capital Y, small y. Then again capital R, small r, capital Y, small y, capital R, small r, small y, small y. Then again capital R, small r, capital Y, capital Y. Then uh, capital R, small r, capital Y, small y. Then again same small y, small. Try to do practice at home whenever you get the time. More you will practice, more you will learn. So then small r, then capital Y, then small r, capital Y, small y, then small r, capital Y, small y, then capital R, small y, small y. See, these were the things. So now we'll make it more clear. Okay, we'll just type fast. So capital R, capital R, capital R, capital Y, capital Y, capital R, capital Y, small y, capital R, small y, small y, capital Y, small y, capital R, small y, small y. Again, capital R, small r, capital Y, capital R, small r, capital Y, small y, capital R, small r, capital Y, small y, capital R, small r, small y, small y. Again, capital R, small r, capital R, small r, capital Y, capital Y. Okay. Then capital R, small r, capital Y, small y, capital R, small r, capital Y, small y, capital R, small r, small y, small y. Okay. Again, what we found? Small r, capital Y, capital Y. Small r, capital Y, small y. Small r, capital Y, small y. Small r, y, y, y. Okay. So these are the 16 traits we have found, the 16 progeny we have found in the ratio. And in this, we are going to see which one is, see, now we will see that which one is, see, this one is dominant one. See, this one is dominant one. So how much we are going to find? Now we will find the genotypic ratio and the phenotypic ratio. Okay. So we have this one. Okay. So this one will be how many we are going to find? The tall the tall plant and the yellow seeded. No, not tall plant. How many of you are first we have to count the character. How many of round yellow is there? So how many round yellow is there? We will just write it here. How many round yellow is there? How many round yellow is there? So this one. So this one is round yellow. Okay. This one is round yellow. This one is round yellow. This one is round but not yellow. Then again, this one is round yellow, round yellow. This one is not. This is round but not yellow. This one is round yellow, round yellow, round yellow. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So how many round yellow we have found? We have found 9. Okay. Again, how many round green is there? How many round green is there? Round green. So this is round and this one is green. This one is round, this one is green. This one is round and this one is green. So how many round green is there? 3. Again, how many wrinkled and yellow? Wrinkled and yellow. Wrinkled and yellow. How many is there? So wrinkled. So this one is wrinkled yellow. This one is wrinkled yellow. This one is wrinkled and yellow. Okay, this is wrinkled and yellow. So how many wrinkled yellow? 3. Okay. Again, we are going to find how many wrinkled green. So this one is wrinkled and this one is green. So this is 1. Okay. So how many total we have taken? We have total 9, 3, 12, 12, 3, 15. Total 16 progeny in the dihabitus was found, in which 9 were round yellow, round green 3, wrinkled yellow 3, wrinkled green 1. We have found in dihabitus, in which we have found, because everyone is dominant one. See, here the round is dominant and the yellow was dominant. Round was dominant, and here you can see that capital Y small y, in which the yellow is dominant one, which has suppressed the expression of the recessive one. Then again is the round yellow, round yellow. So like that, the phenotypic ratio was 9 is to 3 is to 3 is to 1. So what in the phenotypic ratio, what we have found? In the phenotypic ratio we have found the 9 ratio 3 ratio 3 ratio 1. But what about the genotypic? What about the genotypic? How many genotypes we have found? How many? So how many genotypic we have found? Okay, see. Okay, this one, how many we have found? This one, only one. Okay, only one, only one. After that, how many we have found? This one, we have found one, total two. Okay, only two. Okay, then again, this one is only one. Okay, again, one, capital, then again two. Okay, two we have found. Then one, two, three, four. Okay, then like this, how many we have found? One, two. Again, one. Okay, then again one, two. Then how many found like that? Okay, so now we will discuss how many it is there. So just count the same color. One, one is two is two, 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 1 is to 4 is to 2 is to 2 is to. So just count how many we have discussed these things. Like we found 1, comma 2, comma 3, 1, comma 2, comma 1, comma then 2, comma 4, comma 2, comma 1, comma 2, 1. Okay. See, 1 is there for this one, then 2 is for both this one, then again this is the only one. Capital S only is only. This one is only 1. So 1, then 2 for this 2 1, then 4 for this 4 1, then 2 for this 2 1, again 1 is only for this one, then 2 for this one, and 1. So the genotypic ratio will be 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So these are the genotypic ratio we have found in the dihybrid cross. See, these are the dihybrid cross. So Mendel has taken first in the monohybrid cross. After then he have found that dihybrid cross, in which he have found that in which I found the genotypic ratio is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. But the phenotypic character, that is 9, he has found the round yellow, the 3 he has found the round green, and the 3 has he found that is wrinkled yellow, and the 1 he has found that is a wrinkled green, and he has found only one recessive character of the those, that is the 1. So that is the wrinkled green. So these were the dihybrid cross of the Mendel. Okay. So what is the so same things they have discussed here, like the round green, then he has crossed the round green and he found these things. So first he has done the two character, first he has done the mono hybrid cross. After that again he got the F1 generation. From the F1 generation he again found that from the F1 generation again he has got for the dihybrid cross. In that dihybrid cross he has found 16 the trait different individuals and the 16 different characters of that both contrasting tra- traits. Okay, so what is the Mendel's second law? So that was the law of segregations. So what is the Mendel's second law? So the law of independent assortment, alleles of different character separate independent from each other during gamete formation. So like you can see here, for example, if so law of independent assortment, like allele of different character separate independent from each other during gamete formation. For example, you can see here the when for in the for example in human beings, we have the gamete like for the M, that is XY chromosome responsible for the male and the XX chromosome is responsible for the female. But after during the gamete formation, what happened? There will be independent assortment, like the both gamete, whatever the allele present in that, both are segregate, like they had independent, they can be independent, they have independent character, they can go through any gamete formation and they are independent, they are going to combine, like not they are not he's that is not going to take the 50% part of this and the 50% of this, but because of this gamete formation, during gamete formation they are individually separated, they are individually sort. After that and any individual can go through any gamete and they can go through the any trait and they can fuse, and after that they can form any gamete. Okay, so uh, these are the law of independent assortment. It is the characters separate independent from each other during gamete formation, and alleles of texture were absorbed independently from those of seed color. See here, here you can see the round green was there. Okay. Okay, you can see here the Alice was independently assortment. How? See, here was round color was the capital or capital R, the capital R for the round color, round shape. The wrinkle, the small r small for the wrinkle shape. The small r for the wrinkle shape. Here you can see the capital Y capital was the dominant yellow, capital small for the green. Then again you can see the yellow, but you can see after the in the crossing what happened, that the character of that seed color or the seed set, that was independent assortment. They are not going to be mixed. Every in the alleles, even the every allele in we found allele in pairs. So the alleles even or the have a separate trait, like they are independently assortment. They are not going to fuse with each other. So that is the law of the independent assortment. Like every character has a separate every they have separate individuals, and they will. So alleles of different characters separate independent from each other during gamete formation, and alleles texture were observed independent from those of seed color. So the texture and the seed color were have shown the observation indifferently, not they are the same type. Okay. Then what are the sex determination in humans? So these were the all about the Mendel's law. Then what is the sex determination in humans? So in humans we found the 23 pair of chromosomes, in which the one is sex chromosome and one perisex chromosome, and the 22 is homologous chromosomes. Okay, so in human beings we are going to find 23 pair of chromosomes in the males and in the females, in which there is one chromosome that is responsible for the sex chromosome. That chromosome is going to determine whether the child is going to be girls or whether is child going to be a boy. Okay, so somatic cell in humans contain 23 pairs of chromosomes. Out of them, 23rd pair is composed of different types of chromosomes, which are named as X and Y chromosome. The 23rd pair contains one Y chromosome in a male. On the other hand, 23rd pair in a female contains X chromosome. This means that all the X would have X chromosome as the 23rd chromosome, while a sperm may have either X or Y chromosome as the 23rd chromosome. When a sperm with X chromosome fertilizes the egg, the resulting zygote would develop into a female child, and when a sperm with YX chromosome fertilizes the egg, the resulting zygote would develop into a male child. Okay, so see, you can see the diagrammatic representation is here. So in human mix, in the male we generally found the XY chromosomes, but in female we are going to found only X chromosome. So anyhow, female is going to give only X chromosome. It is and once whatever the sperm from the sperm, whichever the gamete will, if it will be attached to X, if it is attached to X, so it will it will develop the zygote into a female child, and if it will be Y attached to that X, it will be developed into a that zygote develop into a male child. Okay, so in females, generally in the X, that only X chromosome is present, only one X chromosome present there. But in males, we have a sperm. In a sperm, we have X chromosome or maybe Y chromosome. Whatever the chromosome, the part of that chromosome will attach to that egg chromosome, that will determine whether it is going to be a girl child or it is going to be a female child. So today class we will wrap up here. Like so today in this class we have discussed about the in the earlier class we have discussed monohybrid cross. In this class we have discussed about the dihybrid cross. See, there is one question is there. A man with blood group A marries uh, a man with blood group A marries a man with blood group O, and their daughter has blood group O. Is this information to tell you which of the trait blood group A or O is dominant, Y or Y not? So in this, what will happen? Blood group O is dominant and the A is recessive. See, the blood group a man who has blood group A marries with a blood group O, and their daughter has blood group O. Okay, After marriage, they have children, and their daughter has blood group O. If this information is tell you which of the trait blood group A or is dominant, why or not. See, in these questions, they have shown that blood group O is dominant and A is recessive because blood group O was able to express itself in heterozygous conditions. Heterozygous conditions, it seems, in heterozygous conditions, due to which the progeny had blood group O. Heterozygous condition. See, homozygous condition was like homozygous condition. We have different alleles, like capital T. In allele pair, capital T, that is homozygous. In heterozygous, small t, small t. Like that, O was able to express itself in heterozygous condition, due to which the progeny had blood group O. See, the female has already O blood group. Okay, the female is already a O blood group, and the male has A blood group. But female can't segregate. Okay, because they have already only A blood group. And if her daughter is O blood group, so what will happen? What will happen? So it is O was able to express itself in heterozygous condition. Why? Because only O is going to be merged. But in males, might be possible that it will be different. So O was able to express itself in heterozygous condition, due to which the progeny has blood group O. So this is how the determination of sex is determined in the human mix. So this is all about the heredity. Again, we will discuss the further portion of this chapter in the next class. Thanks you a lot, and have a great day ahead.